Click the bell icon and subscribe to our channel so that you will not miss any classes.
In a typical bisexual flower, as we discussed earlier, there are 4 folds like calyx, corolla, androecium and gynoecium. Now we shall understand how these folds are arranged in a flower. The folds of flower are arranged on a base called thalamus. On thalamus, there is presence of ovary, extended style, and it is terminated in a receptor part for pollen grain called stigma. So this is stigma, this is style, and the basal part called ovary. And all these 3 together called as pistil or carpel, or otherwise it is also called sporophyll. Megasporophyll. In some plants, there may be presence of a single carpel, and the condition is called monocarpillary. And in some, there is presence of multiple carpels, that is more than one carpel may be present, and the condition is called multicarpillary. Again, in multicarpillary condition, their pistils or the carpels may be fused or they may be free. Consider these 5 as a pistils. Consider in a flower there are 5 pistils. If they are present free, independent of each other, then it is called, the condition is called multicarpillary apocarpus. Multicarpillary apocarpus condition. If all these are fused like this, if all of these are fused together and they appear to be as a single, then they are called syncarpus. Syncarpus. Multicarpillary apocarpus or multicarpillary syncarpus. Example for apocarpus are papaver, and example for multicarpillary syncarpus is mycelia. Are the examples for apocarpus and syncarpus. Sorry, it is ulta. Example for apocarpus is mycelia, and example for syncarpus is papaver. Are the 2 examples. Now, this is about the androecium and gynoecium. All the pistils together constitute a whole, and that is called gynoecium. We will be discussing about this further more detail under megasporogenesis. Next, from the base of the ovary or from the thalamus, there arises a stamens which are a part of androecium. So these are the stamens. The stamen consists of a lobed structure called anther and a thread-like structure called filament, and both of these together called a stamen. Both of them together called a stamen, and all the stamens arranged in a flower is called androecium, the male reproductive structure. Next, coming to the non-essential poles, that is calyx and corolla. At the base of this, there is presence of a green colored leaf-like structures, and they are called sepals. And all the sepals together is called calyx. And a very colorful part of a flower, that is called the petals, and all the petals organized in a flower will constitute the corolla. So this is petal, and all the petals together we can call corolla. And these two are essential for the pollination related activities. That is, they are responsible for attraction of insects for pollination. Even though they are not directly involved in reproduction, they are indirectly helping the process of pollination. Hence, they aid in sexual reproduction. Now, let us understand in detail about the androecium and gynoecium structures. Let us understand the structure of male reproductive structure. Male reproductive structure, that is androecium. Androecium is containing individual units called stamen, and they are also called microsporophyll. And stamen is made up of two parts, a lobed structure called anther, in which the pollen grains are produced, and a filament. That is, mainly the main part of stamen is anther, where there is production of pollen grains. And anther, otherwise called microsporangium. Then, in anther, there is production of pollen grains. Pollen grains. Pollen grains carry male gametes, hence it is referred as male gametophyte. It is called male gametophyte, or otherwise it is called microspore. Now let us understand in detail about the structure of stamen, structure of anther, and formation and structure of pollen grains in detail. Now, the stamen is consisting of a lobed structure called the anther, and the anther is attached with the filament, and at the base it is attached to the base of the ovary or to the thalamus. And so now this is anther, and the anther is consisting of lobes, and the lobes are otherwise referred as theca. And there may be single lobe present in an anther or two lobes. Single lobe may be present or two lobes may be present. In majority of the angiosperms, there is presence of two lobes, and the condition is called dithecus. If the lobes, number of lobes, if it is one, then it is called monothecus. If there are two lobes present, then the condition is called dithecus. Condition is called dithecus, and in majority of angiosperms, the type of anther we observe is dithecus. And this lobe is attached with a thread-like structure called the filament. One end of the filament is attached to the anther, and that end is called distal end. distal end. The end of filament which is attached to the anther is called distal end. The other is attached to the base of ovary or to the thalamus, and that end is called proximal end. Now, when we take a transverse section of the anther, when we take a transverse section of an anther and observe under microscope, we could observe two lobes like this. So this is the transverse sectional view. Two lobes are present and it is separated by a tissue. So this is one lobe, this is first lobe and the second lobe. The two lobes of the anther are separated by a tissue called the connecting. And in each lobe there is presence of two pollen sacs. This is 1, 2, 3, 4. That means in each lobe two pollen sacs are present, within which there is production of microspores or the pollen grains. Since there are presence of two lobes and 4 pollen sacs in each anther, it is also referred as dithecous. This is we already discussed. That is, two lobes are present. Dithecous, that is two lobes. Dithecous and tetrasporanginate. Dithecous and tetrasporanginate, where 4 pollen sacs are present. Tetrasporanginate refers to 4 pollen sacs. Now, let us understand how these pollen sacs are developed and what are the protective coverings for the pollen sacs in the anther. Now, in a very young anther, that is when the anther is very young, in a young anther, there is presence of a actively dividing cells called meristematic cells that we already know. In all plants, the actively dividing cells are present at the tip or in the growing parts, and they are called meristematic cells. Meristematic cells. And these meristematic cells undergo differentiation. Not all, some of the meristematic cells undergo differentiation into a special type of cells called archisporial cells. Called archisporial cells. And these archisporial cells further differentiated into archisporial cells undergo further differentiation into primary parietal cells, and primary sporogenous cells. And these two are the two different types of cells from which there is a development of two different and important parts of the anther. From the primary parietal cells, there is development of anther wall. From primary parietal cells, anther wall is developed, and from primary sporogenous cells, there is development of pollen grains. Pollen grains. Now, in anther, all the sporogenous cells are capable of undergoing meiotic cell division. Upon undergoing meiotic cell division, they produces pollen grains, which means all of the sporogenous cells are capable of producing pollen grains. Hence, In anther, the primary sporogenous cells which are capable of undergoing meiotic cell division are referred as pollen mother cells. Pollen mother cells. As we know, male gametes are produced usually in a large number, which means all of the sporogenous cells are capable of undergoing meiotic cell division. So, invariably we can say sporogenous cells are nothing but pollen mother cells. From these there is production of pollen grains. Now, I told from primary parietal cells there is development of the anther wall. And the anther wall is made up of 4 different tissue layers, and they are: the first one, epidermis, endothecium, middle layers, and the tapetum. Middle layer and the tapetum are the 4 tissue layers present in a anther wall. Now, the anther is developing from 2 different types of cells, that is primary parietal cells and the primary sporogenous cells. And from these 2 types of cells, there is development of pollen grains and the wall layers. Now, in next class, we shall understand in detail how the pollen grains are produced and how these 4 tissue layers are arranged in an anther wall and their function in detail.